I'm Dr. Miranda Salisbury, an outreach specialist for the Army Education Outreach Program, AEOP, Internships and Fellowships. And it is my pleasure to host today's session and introduce our exciting series of talks that unites our community to explore STEM opportunities and career pathways. The AEOP I&F Speaker Series aims to build community and facilitate learning. Through this series, we'll introduce you to opportunities within STEM, provide insights into various career paths. Our goal is to help you fully leverage all that AEOP can offer, including masterclasses in the AEOP Learning Hub, the newly announced Leadership Certificate, as well as next step opportunities introduced in each Speaker Series section. Before we begin our session, we do have some exciting news. 4 attendees will be randomly selected during the session to receive an AEOP Pocket Scientist or Engineer. These are cool little gadgets that can help you stand out in your schoolwork or work. And to start, we'd like to engage our community a little bit. As AEOP interns and fellows are conducting active research in labs nationwide, we'd love for you to go into the chat right now and share the location of your internship or fellowship. This is a great way to connect with everyone. You can say what level you're at, maybe what you study. Some of you might be at the same site and not even know it, or be studying the same thing across the country. So it's just a great way to engage today. We will be using the webinar function in Zoom, so you won't be able to turn on your mic or camera. However, please continue to interact with each other in the chat and keep an eye out for updates from our AEOP team. For any questions for our speakers, please at any point put those in the Q&A. At the end of the talk, our facilitator, Crystal, will voice those questions. And now I would like to introduce our amazing facilitator, Crystal Jenkins. Crystal is an undergraduate communications intern with the RIT administration team this summer. Crystal, the floor is yours.
Thank you so much, Miranda. Happy Tuesday, everybody. As Miranda mentioned, we will be taking some time at the top of every one of our speaker sessions to introduce opportunities available to you. Today, we'll hear from Ivory Cheney, who is the Educational Outreach Program Manager at the Army Research Laboratory, Army Research Office, or also known as ARO. She oversees the Army Educational Outreach Program, AEOP, and directs STEM outreach initiatives, including the National Defense, Science, and Engineering Graduate Fellowship Program. Ms. Cheney served 21 years in the Marine Corps Reserves with developments to Iraq and Afghanistan, earning several awards. She holds a master's and bachelor's degree in business administration from Liberty University and a project management certification from Cornell University. Take it away.
Thank you so much, Crystal. So as she said, I'll be presenting to you some next steps from your internship and fellowship program. The DoD has some exciting programs out there that you may or may not have heard of. The first is the SMART Service for Scholarship program. It's available now to high school seniors and those at the undergraduate, master's, and PhD level. And for each year that SMART pays for your education, you get to work for the DoD for 1 year at a sponsored facility of your choice. Some of the benefits of the SMART scholarship is that they pay full tuition, they pay you a stipend, they give you health benefits, you get a travel allowance, a book stipend, you get internship and mentorship from scientists and engineers from your sponsoring facility, and you're guaranteed a place of employment upon graduation. So as I said, the SMART scholarship does now take high school seniors. The application period opens on August the 1st on the smartscholarship.org website, and it closes in December. If you have more questions, please take the time to go to the SMART website. They have tons of FAQs there that will allow you to find out all of this exciting program and all of the things that the DoD has to offer. Another program that the DoD sponsors is the National Defense Science and Engineering Graduate Fellowship. It allows students to get their PhDs in several STEM disciplines. This scholarship is funded for 3 years. There is no service obligation to the government, and you can attend any U.S. institution that gives PhDs. Just like SMART, some of the benefits of the INDESEG fellowship program are full tuition, travel benefits, a nice stipend, and health insurance, as well as an Army mentor. Both these programs allow students to be able to pursue their educational pursuits full time without worrying about how they're going to pay for cost of living or tuition. They also give you mentorship, they help you to build networks, and they allow students the opportunity to just focus on research and giving back to their communities. I am happy to take any specific questions about SMART or INDESEG if you have any. Some of the eligibility requirements for both programs is that you attend a U.S. Institution of higher learning and that you be a U.S. citizen or have be a permanent resident of the United States. For the SMART program, you do have to have the ability to obtain and maintain a secret clearance or higher, depending on the sponsoring facility that you want to work for or that you or the field you choose. SMART does a great job of giving students opportunities to do internships, and some of their newest opportunities include internships overseas in Belgium, the UK, or Germany. And those go on each summer, and it's just a great opportunity. One last thing I want to say is that I myself am a SMART retention scholar. And what that means is once you start working for the DoD, they also give you an opportunity to go back to school in a STEM discipline. So I start school in the fall and I will go to school full time, and SMART will pay for my education. And once I complete school, I return to my sponsoring facility to continue work.
Thanks so much, Ivory Cheney. These sound like great opportunities, and I'm sure questions will be rolling in soon, which we'll ask at the end of the program. Moving forward, I would like to introduce Dr. Aarti Gautam, the Deputy Director at the Medical Readiness Systems Biology Branch at Walter Reed Army Institute of Research. Dr. Gautam specializes in molecular biology with expertise in transcriptomics, epigenetics, microbiome metabolics, and nutrigenomics. Her research focuses on developing countermeasures for illnesses relevant to the military and supporting soldier health. Dr. Gautam has authored over 60 peer-reviewed publications, 2 book chapters, and more than 250 abstracts, presenting her work at various national and international conferences. She completed her PhD in genetics from CCS Higher Agricultural University and conducted research at Texas Tech University under Rockefeller Fellowship. As a principal investigator, she leads research projects and coordinates a multidisciplinary team. Today, Dr. Gautam will discuss integrative biology approaches to diseases of military relevance, highlighting a holistic approach to understanding biological systems. Please join me in welcoming Dr. Aarti Gautam.
Thank you. Thank you very much for introducing me to the group. Can you all hear me, first of all?
Yes, we can.
Perfect. I would like to introduce myself here working in a DoD setting. And what I do is basically working on various diseases of military relevance. And please feel free to interrupt me, ask question in between. I want it to be an informal discussion today. I just want to give you a perspective of my journey from where I come and how and where I'm proceeding. So please feel free to reach out to me with any questions you may have. Just write in the chat, please. So I work on in a DoD setting, and we work on most of the diseases that are relevant in military settings. I have to, this is just a disclaimer slide, and I would definitely like to start my talk with who I am. And I want you to be aware that this is the most important part of me. And I was born and brought up in India. And my parents, they raised me and my siblings, and they empowered us to dream. And they empowered us to dream big. And they also provided us whatever they could to fulfill our dreams. And I am really thankful to my parents and my family back in India, who has always supported us. Currently, I'm in Maryland, and I am working at Walter Reed Army Institute of Research. And I definitely enjoy working in the DoD setting. And here I'm living with my family, my husband, and my two beautiful kids. So to give you a little bit idea about my journey, as I have mentioned, I grew up in India. I did my high school. When I was doing my high school, I had really no idea how I wanted, what I want to become in future. You know, that's the question everyone asked. And I had at my high school, I had no idea what I'm going to pursue. So, but I definitely my family, they were raising me at a level that definitely I have to do higher education, but I did not have a direction there. I knew that I will study, I will go for higher studies, but what is the direction? I did not know until unless I did my undergrad. During the undergrad, I did physics, chemistry, biology, and of all these courses, I realized that I have a preference towards biology. And from there, I started, I pursued my master's in science in genetics, and everything was done back in India till I joined a PhD course where I was doing my coursework, all the courses in India. And I wanted to study abroad, and I wanted to try leading labs to learn new things, and to go to one of the advanced labs. So I was applying to various scholarships, and I was lucky enough that I got a fellowship from Rockefeller Foundation, and I came here to Texas Tech University to complete my research. And my research was done, and there I was trained to understand about genes and proteins, and how we can change genes, how we can change the nucleotides that we all have. So I got to get the advanced training here at Texas Tech. I went back to India, did a couple of years of a postdoc at international organization, and sometime some training at a pharma company as well. And then I came back with my husband to do postdoc at Tulane Primate Research Center. And here, when I came to Tulane University, I was doing Lyme disease pathogenesis. And here I got trained to do bacterial pathogenesis. And I was looking into the humans, how Lyme disease infects human, and how we can study tick, and we can study the bacteria, and we can study human, and how they all integrate to find new diagnosis. And one of the labs where I was working, they were advanced to one of the peptide, which was used as a diagnostic kit for dogs. So definitely, I was working in one of the advanced lab there. And then from my postdoc, I transitioned to MRDC, and it's been 15 beautiful years with MRDC for me now. So give you a little bit preview about the science. So the science comes in, you know, we can have multiple definitions of science. And I am here on my slide, there are some of the branches of the science, and they can, here I'm mainly talking about the natural sciences, which comprises biology, physics, chemistry, geology, and astronomy. And I definitely have added in computer science in this, because these days nothing is complete without computers, or nothing is complete without data. So I added it here, and that's where the science can go to any of these branches. And sometimes many of these branches, they integrate to work to find an outcome for any problem. So while working as a scientist, you all may be familiar that what is a scientist? You know, a scientist is a person who systematically gather information, and they do research, and they do evidence-based research, or evidence through literature, and then they create a hypothesis. Once the hypothesis is created, it is used for testing. We test it using in vitro system, animal systems, preclinical. It could in biology, it could be that, or it could be some structural predictions. So we do some kind of a test for every site. It could be a scientific field. It could be different, but there is a testing involved everywhere. And once the results from that testing is obtained, we get new information that is used and shared for understanding or fine tuning, or it can help in the knowledge for other people who are working in the field. So basically, you research, you discover, you transform, and this cycle or this chain goes on in the field in a life cycle of a scientist. So coming to my MRDC research, this image I would like to mention is from a journal article or a journal front page where we propose that a human being is a system of system. So if I have a problem, if I have some issue with me, it's not only showing it by all the entire tissue, my heart, my spleen, my liver, my lung, everything is working together to create that problem. So we consider that and not only that, the surroundings are added factor. So there is an intricate relationship between all these complexity, and we have to look into all of these systems or all of these cells at the microscopic level, and then see what the organs are creating or organs are forming, and eventually resulting to the outcome. What is the outcome, whether it's a happy outcome or a sad outcome, but definitely all the systems, they do interact for an outcome. So I come from a branch called Medical Readiness Systems Biology, and we do a multidisciplinary research, and we try to find solutions for military health, and they can improve military health or performance and the readiness in the field. Our vision is that we are able to integrate different types of data science or different types of biology at a systems level, or maybe in the current arena, synthetic biology, by creating a new system and using artificial intelligence for precision care. And this is mainly using for the diseases that are relevant to military. So the lab from where I am, we do work on bench, and we do bring the solutions to the bedside for the clinics as well. While I'm talking about the bench, my lab is involved in creating animal models. We created an animal model, a mouse model for post-traumatic stress disorder or acute stress disorder. We do work with mouse or ferret model of blast exposure or traumatic brain injury. We have worked with animal models of pain and many more. So we worked with animal models. It could be for different diseases. And what we do is that we collect, once the model is created, we collect their tissues, we collect the multi-omics data. The multi-omics data would be the data that is collected from DNA, the data that is collected from RNA, the data that is collected from proteins, metabolites, or microbiome. So we collect all those data, plus if we are working with the clinical samples, we do get clinical information, psychological information, their physiological parameters, their temperature, body temperature, or blood pressure, or psychological issues. And clinical would be the blood count. So we do get all that data. We integrate that data, and then we try to identify some source, some outcome, how these molecular factors are affecting the outcome of the individuals, or in case of animals, how they are changing the behavior of the animals. And once we do that data analysis, we do diagnostic or prognostic biomarkers. And here we use computational. So all the data that is used, that is generated, we generate tons and tons of data from all these multi omics data set, and then we try to integrate it and use machine learning and artificial intelligence to find the outcome from that data. So our lab has various capabilities, and I would like to introduce a few of that to you all. What we do is, I did mention we get human clinical samples as well as animal models. So I did explain about animal models, but we do work with human samples, such as we do get samples from patients who got PTSD, or we do get samples from the trauma exposure, traumatic brain injury samples. We do get samples from soldiers who are exposed to burn pit toxicants. We do get samples from the soldiers who get exposed to other psychological issues as well. So we get those samples, and our lab has a robotic system, and we do process these samples in-house using next generation sequencing. So when we are talking about the DNA-based assays, we do look into the total content of the DNA, what this human being or what this mouse model or animal model, what is their total DNA content citing to. So we do an assay for total DNA content, or RNA, what is the transcription, or how much data, or how much sorry about that. So how much DNA is getting transcribed, or in a disease state, what is the current expression level of the genes that is there. We do RNA analysis, we do PCR analysis, we do analysis at single cell level. So in blood cells, they are in blood, we have neutrophils, we have lymphocytes, we have granulocytes. So we can tease apart all those cells, and we do look at each of those cells separately. If somebody is exposed to a trauma, what their lymphocyte is saying, what their neutrophils are saying, what is granulocyte is saying. So we do look into every single cell RNA DNA analysis. And we do look into the proteins. If there are any proteins which are being changed, and there are multiple assays, we do that. We look into 1,300 proteins in one assay. So, and some of the assays are targeted. Sometimes the targeted assays are inflammation related. So we can do 100 to 200 proteins in one assay and detect, okay, if this person has some neuroinflammation going on. So we look into their brain assays, and we check their protein, what inflammatory cytokines are being changed, or anti-inflammatory cytokines are being introduced. So we can do that in our lab in-house. And as I told, microbiome, we look at the fecal samples, or we look at the skin swabs or saliva samples, and we look at their microbiome, and and then we study the microbiome content. And though all these microbiome sample type, they lead to some kind of the microbiome is producing some kind of an enzyme or some kind of a metabolites which is playing role. So we do prediction, or we do target identification of all the microbiome which is there in our gut. So gut-brain axis is a big issue when a soldier goes through psychological trauma. You know, you all it's normal that when we are stressed, we eat more, or sometimes for some people, when they're stressed, they don't eat. So and everything is related to the microbiome, what is being expressed, or what is being dominated at that time when somebody's stressed. So we look into that as well in our lab. And all in all, all this data, you generate so much of a data, and everything has to be compiled, integrated, and you need computational solutions. So our lab has a team of data analysts, though they do work together, and then they help us finding solution, and they integrate the knowledge which is derived from all these assays, and we do a biomarker discovery or identify new targets based on the data. So I did mention about our efforts on microbiome, and must tell you, microbiome is affected. Everyone, we eat differently. We are surrounded by different, in a different household. Everything is affecting our microbiome. Some people have pets, some people don't have pets. So the people who have pets, they may have a very similar microbiome to their pets, but sometimes the stress, stress causes microbiome changes. We have published studies like that. If there is a chemical exposure, that will change microbiome. If there is soldiers who are on MREs, their microbiome would be very different from the person who is eating a regular food. So microbiome content changes with every little thing that we do. So, and some of the projects that we are running in our lab are related to nutrition, because we did a study where we tested omega-3 diets and try to observe that if they can be beneficial to the traumatic brain injury study samples. And we did study microbiome in case of radiation, because we get exposed to radiation, or if someone gets exposed to radiation, sometimes like cancer patients, they are exposed to radiation. So how their microbiome changes, because they cannot eat food, and that changes their microbiome, or the medication, it changes the microbiome. So we look into things like that. And one cool study that we did was we are one of the DoD labs who were able to work with NASA to send our samples, or send some of our mice to International Space Station. So when the mice went to International Space Station, we collected their fecal samples. With the help of astronauts, they collected fecal samples for us, and they sent us back, and then we received the mice back. So we did a study on microgravity and have published multiple papers related to microgravity effect on the host. And microbiome is affected. Stress definitely does affect microbiome. Pain, chemical toxicants, and any type of environment, it does affect microbiome. So we are a big supporter of microbiome work, and we do our lab is a big leading lab on many microbiome related projects. And just to give you a little glimpse of our lab. So as you know, whenever we are working on a military relevant issue, I myself as a lab cannot do everything. We have to collaborate with multiple organizations because sometimes people have access to samples, people have access to some of the technologies that we may not have, or the way funding guides us. So we have multiple projects which are in MRDC. So we are working with the ICD, which is in Maryland, on some of the chemical agent exposure. So we worked on some of those projects, and we are also working with the RARE on, in my building, we are working with other divisions on directed energy efforts. We are big time working on sleep deprivation. What happens if a person is deprived of sleep? And at RARE, when I'm saying RARE, that's my institution. At RARE, we have a sleep study lab, and there they are doing multiple experiments. So we are working with them to identify what is causing what genes or proteins or metabolites may be causing, because changing with respect to the circadian rhythm. So we are looking into that. We are working on multiple institutes with systems biology projects on coagulopathy or burn, radiation, wound, and tissue oxygenation. And our big project is with Usarium, which is in Boston, NATIC. Massachusetts, about nutrigenomics and physical stress and austere environments. What happens if there is a high altitude? What happens if there is a heat stress, or what happens if there is a cold stress? So we get those samples from soldiers or animal model, and we do team up with them to assay and find some answers for each of that. Not only MRDC, we work with other government agencies as Navy Research Lab, Army Research Labs, and Air Force, and so pain clinic. We do work with NASA, I did mention, and we do work with the Naval Medical Center. We do work with the Armed Forces Radiation Institute and Uniformed University, as well as NIH, NCI for multiple of projects. And many of the projects need industry partner or academic partner. So we team up with academic partner for one of our PTSD project, where we are teaming up with 6-7 different institutes to identify some biomarkers for PTSD. And these biomarkers can aid in presentation or aid in diagnosis of PTSD. So currently the PTSD is only diagnosed using a questionnaire, and that's the subjective way. And we want to help by looking at the blood samples and telling that, okay, this person may have a chances of developing PTSD in future, or this person is in a subtype or in a different subgroup that may be getting PTSD. So we work in different populations like that. We work with so our team also works with burn patient, and when we get the burn patient, we have developed a panel of gene expression markers, RNA-based markers, where we can mention that these patients will develop PTSD, or sorry, these patients will develop sepsis in 4 to 5 days after sample collection, or we can predict within 24 hours that these burn patients will develop sepsis. So we have developed one diagnosis panel, and we have patented that. So just to give you a little glimpse of our collaborations, and our research is focused on multiple of these issues. And to give you some deliverables that we have produced from our lab. So as I was explaining about the PTSD biomarkers, so what our lab has done, that we worked on 5,000 like different cohorts. And when we are working on different cohorts, some of them are active duty, some are veterans population, some is civilian. So we try to gather so many different cohorts and data from all these cohorts, and eventually combined all that data. Every data has a uniqueness, but sometimes because of the way the cohort is, the PTSD signal is diluted. But our computational analysts, they really worked day and night to develop some biomarkers, and we are in a process to send this to FDA for commercialization, and we are working on doing a pre-submission for FDA for this particular project. The second is a panel of predicted biomarkers for sepsis. So in case of burn patients, what we did was we collected samples from we collaborated with Burn Center, which is at Washington Hospital. And as soon as somebody experienced the unfortunate event of burn, they came to emergency department, and the sample collection started as soon as they were admitted to the emergency department. And we started collecting samples after every 2 hours, and the samples were collected for at least for 21 days, and sometimes more if the patient was in the clinic for a longer time. So we had the data points for every 2 hours for many of these patients, and we did look at the gene expression markers, and we did identify some predictive markers of sepsis, like within 24 hours that, and these, and eventually we knew from our data, we knew that those 4, 5, or those markers, the 5 genes that we are looking, if they are changing, this person will develop sepsis in 5 years, 5 days down the line. So we have diagnosed a predictive marker for sepsis in case of burn patients. We have also identified some of the microRNA and metabolites for radiation exposure. So when someone is exposed to radiation, what is happening to their microRNA and metabolites. So we have done that, and right now we are in a process of patenting that as well. In case of acute mountain sickness, this is a very neat study. So soldiers, we collected the blood samples from the soldiers who were at the sea level, and the same soldiers then experienced the peak spike, which is one of the largest peak in United States. So the soldiers marched there, and we collected samples from their from the post hike, and once we have that the blood samples, we compared their sea level samples to their mountain mountain. After reaching mountain, we collect samples collected post mountain hike, and we compared the samples, and then we had the physiological markers. After doing all the playing with all the numbers, we have developed a panel of blood markers, which again is being patented, and we are working on developing a pharmacogenomics tool. So to give you an example, like how I am different from my husband, or how I'm different from my son. We all are in the same house, but one of us, and when we are exposed to some kind of cold, I behave very differently than my husband, and my kids behave very differently. So what is different between each of us? What DNA, what genes are different? What single nucleotide polymorphism are there? So we have developed a small kit which can detect the single nucleotide polymorphism, and this is just using your spit. You spit in that and in that kit, and it gives you what is changing. So and this is basically something very similar to the COVID test that we all have done a few years back. So this pharmacogenomics tool can be used for any disease. Like it's disease agnostic, and we can utilize it for anything. Wherever we know that there is a change of nucleotides, this tool can be used. So besides that, these are some of the clinical products we are working on. And we are also working on many of the knowledge product. Our lab publishes papers, manuscripts on routine basis. Feel free to look for my name, and you will see some of them. If you're interested, please reach out to me. I'll be happy to answer any questions. And we are working on some immune related to chronic stress biomarkers as well. So we have a multitude of knowledge products from our lab. And when extreme right, I'm talking about some small business technology transfer efforts. So what we do is we collaborate with some of the industries, and whatever we are identifying, I identify a DNA that I think is changing in case of a disease A. Now, what will happen? Like, we need to translate that product. So we do work with some small industry partners, and we do work with them to make it the field, so that we can make those product usable in field. So we have multitude of STTR efforts that are going, and where we are trying to associate our findings to those products that the company has, and making some kind of a device which can be easily transported to field, or easily interpreted, or applied to role of care 2, 3, wherever applicable. So we are working with industry as well on translation of products. And I'm telling you all about what we do, but everything comes with some kind of a roadblocks. And we are affected by funding and evolving technologies. With current new technologies with 3D printing and with all the data analytics platform coming, every day there is a new technology coming. So does that mean that we don't trust the old technologies? And how that should not be the case. So how we can use the new technologies to how we can adopt them to evolve, like that's a big learning curve for everyone. I think you all will be facing that in future as well. Things change on daily basis. How you can utilize or how you can use these new technologies to give a better product. So that is something we face every day. Reliability and reproducibility. When I'm talking of animal models, how reliable are animal models? I'll be working on an animal model for PTSD or animal model for traumatic brain injury. How good is that model if I have to translate that product to clinic? We have to do a lot of validity. We have to do a lot of refinement to make sure these are reliable. So definitely sometimes with the sometimes like you cannot get brain samples for a creature, for people who are experiencing PTSD, because there is no way I'm going to get any samples. So how I can make reproducible results using my preclinical model, what I can use. So looking into other factors, that is important. Ethical considerations definitely come into being, and complex. Sometimes you have to work with different, like I am biologist. I don't have 100% knowledge of physical science or chemistry. So you have to have complex collaborations to get to understand their perspective, and that adds to challenges sometimes. So definitely that's a part of the game, but you do get success in every game. But whatever I would like to say, that whatever I do, if anyone you are listening, if you want to become a scientist, curiosity, that's the key thing. Always be curious about what's going on, how you can make things, how you can change things, or how you can make things better. So and it comes with a lifelong setting of learning. It's a learning curve. Every day I learn. Like it's not that the learning phase has stopped for me. I learn every minute here. And sometimes with you get setbacks, and you have to embrace resilience as well. And every failure, it comes with a learning as well. So be patient. That's the way I have been taking my career, that I be patient and keep a positive mindset, which is not always possible, but that's the way one should pursue. And I think that I would like to end my talk here, and would like to give you where I stand at MRDC. So I am at WRAIR. I am at Walter Reed Army Institute of Research. And I come from a branch which is CMPN, which is Center for Military Psychiatry and Neuroscience. And there I stand at MRDC. And that's all I have. Thank you.
Amazing presentation, and thank you so much, Dr. Gautam. I know I've learned a lot, as well as our participants, and I will ask some of our questions now. One of the first questions that was asked in the chat was, if you were asked to persuade an undecided college student to study biology, what would you tell them, and why should they follow your academic track?
If I have to tell someone, for me, I can only talk about me and the folks whom I talk. It came from inside. When I was doing my undergrad or grad, I had no idea what I want to become, and I really had no idea till undergrad. And at sometimes it's the way the position things are aligned for you. You get that direction from the way things are aligned for you. And you have to, the first thing is you have to like what you do. That's the most important thing. And I always enjoyed biology. Whenever there was a chemistry related project, I enjoyed it, but it was not the best thing. So it's the way your mind and your soul guides you. Basically, you follow that path, and and whatever it is, that should make you happy. That's the most important thing, that I do that. I enjoy my work. Doesn't matter if I work 10 hours a day, I still enjoy it. My family gets really upset with it, but I enjoy my work.
Thank you so much. So the next question we were asked was, when creating an animal model for a study, where do you start, as in how do you know what to change in order to create an effective model as possible?
Yes. So when we are working on animal models, we do, first of all, you consult literature. You look into what's been published and what are the leading labs who are doing it. And then you consult the people who experience those problems. Sometimes because when I'm working with PTSD, I had to know what my soldiers face. So I talk to them verbally, and I tell, okay, they experience sleep loss, they experience anxiety. So I have to make sure all those parameters are reflected in my animal model. So when I create an animal model, if my model is not showing any of those outcomes, that's not a good model. Definitely whatever clinical whatever is seen in clinic should be reflected in an animal model, and you definitely need to consult the people who are working in the field, people who have experience in the field, clinicians, as well as all the literature that has been published in the field. So it is a lot of startup material that you have to look into before you plan on anything like that.
Okay, and one of our final questions is, what is the most exciting or interesting part of the research that you have conducted?
You know what? I have to tell you that I was a biologist, so I have always worked in tubes like DNA, RNA mixing together. I always working with tubes. The exciting part was when in the recent times, whenever we generated this data, and I'm working on this data, and I send this data to our data analyst, and they used to talk about okay, the output, that this is they integrated it. They used to talk about algorithms, and they used to talk about the analytical platforms that they have been using. And I used to be really blank there what they are talking. I never learned their language, like what they were talking. So I was really curious, and in past few years back, what I did to myself was I enrolled myself in a bioinformatics course. So I spent evening times doing that course, and once I did that, I felt very happy that I'm glad that I did that, because I learned other person language, and that added extra kind of information, because I think from a different person perspective as well. So that that made me very happy when I did that, and I'm able to learn. I'm able to understand other people's language. I think that's something I enjoyed, and I like it.
Thank you so much. I will give you a round of applause on behalf of all of our participants. We would like to thank you, Dr. Gautam, as well as Ivory Cheney, for your amazing presentations. We appreciate everyone here for attending our first ever AEOP Internships and Fellowships 2024 Speaker Series. Our team at AEOP is always excited to hear and see all of your stories, so make sure to send in a picture from wherever you're watching in order to win some free merch. You can also DM us your photo on social media under the username AEOP I&F, and that is in the chat. Thank you once again for joining us. Our next webinar is July 30th from 3 to 4 p.m. featuring Dr. Roy Waterman from University of Vermont, who will be presenting on essential skills to take control of your time and manage various demands efficiently. We hope to see you all there. Have a good evening, everybody.